and it actually can inhibit selected genes. It actually can turn genes off. So when the fat insulin receptor gene was turned off in mice, these mice ate ravenously and remained slim and got the health benefits of being slim. They didn't get diabetes. They didn't get heart disease. They lived 20% longer. They got the benefits of caloric restriction without the restriction. Every major disease and every major aging process has different genes that are used in the expression of these disease and aging processes. And being able to actually selectively turn them off is one powerful methodology. We have the ability to turn enzymes off. So one drug, for example, that's in phase 3 trials, torcetrapib, turns off a key enzyme that destroys the good cholesterol, HDL, in the blood. If you inhibit that enzyme, HDL levels soar and atherosclerosis slows down or stops. There are thousands of these types of developments in the pipeline. 99% of the drugs on the market today were not done that way. They were done the old way, called drug discovery, which we just happened to find something that did something beneficial, like, say, lower blood pressure in someone who has hypertension, but we had no idea how it worked or why it worked. And invariably, it was a very crude, these are very crude tools that have lots of side effects and complicated effects. The new paradigm of rational drug design involves actually understanding these information processes, the exact sequence of steps that leads up to, let's say, a process like atherosclerosis, which causes heart attacks or cancer or insulin resistance that underlies type 2 diabetes, and providing very precise tools to intervene in these processes. And our ability to do this is also growing at an escalating rate. DNA sequencing came down from $12 to a penny in 1 decade. The Genome Project was very controversial when it was first launched. The mainstream scientists said, there's no way you're going to be able to do this in 15 years. We just had our most advanced PhD students and our best equipment, and around the world we finished 1/10,000th of the project. There's no way it's going to happen in 15 years. And 10 years later, the skeptics were going strong, saying, I told you this wasn't going to work. We're now 10 years into a 15-year project, and only 2% of the project is finished. But it's the last 7 doublings that go from 1% to 100%. So the explosive nature of an exponential process that basically gets most of the project done in the very late stages is phenomenal. This graph shows you basically doubling the amount of genetic data that we have collected each year. And there's many different ways to look at biology, but our ability to understand it and to change these disease and aging processes is doubling every year, which is very powerful exponential growth. Communication technology is also doubling in many different ways: capacity, bandwidth, price, performance. The size of the internet has doubled every year. So in my first book, I had a little bit of this graph. There was only 10,000 nodes in the internet, serving a few thousand scientists here at Harvard and MIT and a few other schools. It was doubling every year. It was clear to me that 10,000 would be 10 million 10 years later, going to 20 million to 40 million, and would be on everybody's radar screen. So if you look at the same data on a linear graph, it looks like the internet came out of nowhere in the mid-1990s, but you could see it coming if you looked at the inherent exponential nature of the process. Another exponential process is miniaturization. We're shrinking technology at a rate of 4 or 5 per linear dimension per decade. These are some animated illustrations from Eric Drexler's book, which was done at MIT in 1986, that laid the foundations for nanotechnology. And we actually have now little systems that are operating at the molecular level. In this next book that's coming out this fall, The Singularity Is Near, I describe many projects where we're building little robots. One actually is a robot that walks with a human-like gait, but it's built at the molecular level. So we're showing the feasibility of actually constructing things at the molecular level that can perform useful functions. And one of the biggest applications of this, again, will be in biology, where we'll be able to go inside the human body and go beyond the limitations of biology. And that brings up a key issue, which is that biology, although it's remarkable in its intricacy and its cleverness, is also very limited, because an evolutionary process has to build things with assumptions it made earlier. So, for example, out of proteins, which is a very limited class of substances. They're three-dimensional, but they have to be folded up from a linear sequence of amino acids. So as we're actually reverse engineering biology, and one of the things we have reverse engineered is red blood cells, we find that we can design them to be thousands of times more efficient than their biological counterparts. So this is one design done by Rob Fried for a nanorobotic red blood cell. It's actually a fairly simple device. It just stores oxygen, lets it out. A conservative analysis of these robotic respirocytes shows that if you red blood cells with these robotic versions, you could do an Olympic sprint for 15 minutes without taking a breath, or sit at the bottom of your pool for 4 hours. So it'll be interesting to see what we do about these in our Olympic contest. Presumably we'll ban them, but then we'll have the specter of high school kids routinely outperforming the Olympic athletes. This is a design of a robotic white blood cell, a little more complicated. It downloads software from the internet to combat specific pathogens. If it sounds very futuristic to download something inside your body to perform a health function, I'll point out we're already doing that. There are about a dozen different neural implants that are either FDA approved or approved for human testing. One that is FDA approved for actual clinical use is an implant that replaces the biological neurons destroyed by Parkinson's disease. And the neurons in the vicinity of this implant then receive signals from the computer that's inside the patient's brain, and it's perfectly happy to get signals from a computer, whereas it used to get signals from biological neurons. And this hybrid of biological and non-biological intelligence works perfectly well. And the latest generation of this neural implants allows you to download new software to your neural implant from outside the patient. Now, these are devices today that require surgery to be implanted, but when we get to the 2020s and with the miniaturization I've talked about, we will ultimately have nanobots, which is really the killer app for nanotechnology, which are blood cell-sized devices that can go inside the body and brain to perform therapeutic functions, as well as advance the capabilities of our bodies and brains. If that sounds futuristic, I'd point out that we already have blood cell-sized devices that are nano-engineered, working to perform therapeutic functions in animals. And some of these are now approaching human trials. There's one that's blood cell size, actually cures type 1 diabetes in rats, lets insulin out in a controlled and intelligent function, blocks antibodies because type 1 diabetes is an autoimmune disease. So these are already beginning to work. When we get to the 2020s, that really will be the golden era of nanotechnology. And these little nanobots will have computers in them, and they'll be able to aggregate their intelligence on wireless local area networks and so on. If we return to the exponential growth of computing, there's the 49 computers from the key computers on the 20th century. By 2020, $1,000 of computation like this, although it won't look like that anymore, will be comparable to the computing capacity of the human brain. But the key issue really will be, will we have the software of intelligence? When I wrote in 1999 that we will ultimately have at least the hardware aspect of human intelligence by the 2020s, that was controversial. I would say it's not really controversial anymore. That's pretty much a mainstream view. The controversy, or the more interesting question, is will we have the software or the methods of human intelligence? And for that, the key issue really is reverse engineering the human brain, something I talked about quite a bit in this book coming out this fall. And there also we're making, not surprisingly, exponential progress. The spatial and temporal resolution of brain scanning is also doubling every year. We didn't actually learn much about the brain up until recently through reverse engineering because we really couldn't see what was going on. Things like fMRI are very crude. You have very broad spatial resolution. You couldn't see signaling on individual interneuronal fibers. The latest generation of scanning technology actually allows us to see in vivo, in a living brain, signaling on individual interneuronal fibers in real time. So we can actually see what's going on. We can see our brain create our thoughts. We can actually see our thoughts create our brain. Because as we think about something, we create new spines, new dendrites, new synapses, and so on. Now the next question is, okay, we can get this data. We're starting to have very high resolution brain scanning, but can we make any sense of it? Maybe it's just an overwhelming amount of data and too complex for us to understand. Doug Hofstadter, Pulitzer Prize-winning author, says, well, maybe our intelligence is just below that threshold needed to understand our intelligence. And if we were smarter and able to understand it, well, then necessarily our brains would be that much more complicated and we'd never catch up with it. But it turns out that that's not the case. We do have actually a lot of data on several dozen regions of the brain out of the several hundred that exist. And we're finding that we are able to express